All right. Thanks everyone for coming to week 2 of our graduate student talks. It's great to have another great turnout again this week. For this week, we have up first Israt Jahan. Before coming to WashU, Israt received a Master's of Science from IIT Bombay, where her thesis work was in molecular parasitology and secondary endosymbiosis. I was really worried I wasn't going to be able to pronounce that. You're currently a 6th PhD student in the Strassman-Queller lab. Israt received a 5-year PhD fellowship from McDonald International Scholars Academy. In her free time, Israt loves fish, both raising them and eating them, but not eating the ones that she raises. So, with that said, let's give it up for Israt.
Thank you, everybody. Is this audible? Yeah, okay. Thanks for coming. The title of my talk today is this question of whether unselected incompatibilities can drive the origin or maintenance of aggregated multicellularity. And because that's a bunch of jargon, the other part is that I'm revisiting the egg problem in the evolution of multicellularity. And before I begin, I would like to ask all of you, what's common between all of these organisms? And I don't have too much time, so they all start life from a single cell, that is one fertilized egg. And this is true for most of the organisms that you see around yourselves, plants, animals, a lot of fungi. They often start life from a single cell, which undergoes multiple divisions, such that all the resultant cells in this organism are clonal in nature. And any sort of conflict or cheaters that can arise, any sort of genetic conflict that can arise in this organism is from mutation within the organism and not external joining of other cells. So what the single cell bottleneck here does, the egg, is that if you have a mutant bad egg, it gets chucked out of development because it's poor. But the question is, why not a multicellular bottleneck? All these organisms that are very diverse, that are very sort of occupy a huge range of environments. So why have they all always evolved this one particular developmental pathway? And the answer that has been for the field for a long time is of kin selection, which is an adaptive explanation, where it's beneficial for these cells to cooperate into an organism because they are highly related. So as your relatedness here goes higher, your benefits also increase because it's a product of the benefits. And because multicellularity is a costly task, so it's beneficial to do it with your kin. Whereas there are other explanations, one of them primarily being by Woolpert and Zasmary in their paper, where they said that it's not primarily a question of kin selection or competition, but it's an issue how the cells develop or coordinate their developmental processes together as they grow into an organism. So here they're basically talking about a non-adaptive explanation, where if you're evolving independently, you could have problems. So this is, I'm calling the unselected incompatibilities hypothesis. So now think about this, cells have to talk to each other during development. And let's imagine one of these cells needed a haircut. So it's talking to another cell saying, are you the nai, which is in Hindi means barber. But if the other cell doesn't speak Hindi and speaks Tamil, to the other cell, it sounds like the other cell is asking, are you a dog? Because they do not speak the same sort of language, so to speak. So essentially, for cells to communicate during development, they would need to speak the same genetic language. And what that means is that when a cell, the rules for gene activation or inactivation have to be the same across all the cells within the organism. And having differences there, especially for very complicated pathways, can have huge downstream sort of consequences, where this is a very complex figure. But the point is, any of these enzymes going into disarray causes one or the other disease, like Alzheimer's or cancerous stuff. So basically what happens is, the solution that evolution has repeatedly come up with is that a unicellular propagule kind of solves both of these problems, where the resultant cells, they are genetically identical, so there is the kin conflict is minimized, but also as they inherit the developmental processes and the program for that, they are bound to have same set of rules that they can follow together. So my question here is whether unselected incompatibilities, let's say before grouping can happen, or selection towards or away from grouping can happen, can such incompatibilities reinforce why most organisms develop from one single cell? And I want to take a speciation analogy here, so I can have a hypothesis I can test. So if you're familiar with, let's say, the Dobzhansky-Muller incompatibilities, where an ancestral population, if it is evolving in isolation, it could sort of incorporate different sets of alleles or mutations that may not be compatible when these two isolated populations re-encounter each other. So in this case, a hybrid sort of offspring would be either non-viable or will have lowered fitness. So similarly, if you just think about it in terms of cells that are evolving independently, in an incipient multicellular sort of organism, and while they're evolving separately, if they have to now come together and develop into an organism, this hybrid sort of organism should have lowered fitness if these unselected incompatibilities for multicellularity exist. And what better than microbial evolution to have a whole bunch of organisms evolving in isolation, in huge replicates, and so on. But also, we would need an organism that can develop not just from a single cell. That's how evolution has happened. And because, unfortunately, I can't go back 4 billion years, so we would need an organism that can develop from cells that may not be genetically identical. And lucky for us, there are organisms that are a minority in terms of the mode of multicellularity they take. But these organisms, called aggregative multicellular organisms, are represented in bacteria and also majorly in dictyostelids, where what they do is, there are about 100,000 of cells that are sort of dividing, growing independently, but they can come together under harsh environmental conditions and make an organism. But the thing is, because these cells are neighboring cells that may not be identical, the lack of a single cell bottleneck of having one particular egg or a sperm or whatever means that any new allele or mutation that appears is probably there in sort of intermediate proportions, or is present in one or not present in one. So how does natural selection deal with all of this? But my organism of interest here to test this hypothesis of unselected incompatibilities is Dictyostelium discoideum. What Dictyostelium discoideum is just a fascinating organism where you have all these single-celled amoebae in the soil, and they are dividing, growing independently, but as they run out of food, they can form a mound and come together upon starvation. And this mound then moves towards light or heat, and is called a slug. And it's a sort of a very complex developmental trait that requires coordination between a lot of these cells within the mound that are now moving together towards light and such. And after At the end of the development, these cells, a vast majority of them become the spores, which are held on top of this dead stalk, and these cells will not have a fitness in the next generation. The spores will be dispersed into new locations where they can hatch into new amoebae, and the life cycle continues. And it's really, like I said, the slugs are very interesting, and I can show it to you. So you can see that the slugs can merge together. They can separate from each other. So it's a really complex trait, and it is the trait that I will be focusing on for the rest of my experiments. And like I said, experimental evolution is the sort of strength of these microbial systems, and DICT provides an additional benefit of having this unicellular and multicellular life stages sort of separate that you can manipulate. So in this experiment that was done by my friend, Dr. Tyler Larson, he evolved Dictyostelium to be in vegetative state such that these amoebae were fed with abundant nutrients and had no reasons to enter sort of multicellular development, so they should stay as single-celled organisms. And he did this for a long time, more than 30 transfers, and that's like, but basically the point is, when the lineages that evolved from one particular ancestor, and this was done in triplicates, they acquired mutations or lost adaptations that gives them a sort of different genetic background than the ancestors. And I have used these lines for my experiment. So my experimental treatments are ancestors, the evolved lines that have diverged from a particular ancestor, and a pairwise mix of the evolved lines with their own ancestors. So for these three experimental treatments, I am measuring the developmental trait of average slug migration. And for that experiment, what I did is I laid down spores from these three treatments of Dictyostelium discoideum with food bacteria on starving plates. So this plate doesn't have nutrients and is covered, and light can enter from one end of the plate, such that once these spores hatch into amoebae, they should migrate as slugs towards light. And then I can measure this, measure the distances of individual slugs that have traveled further from this so-called starting line. And I did this for a whole bunch of plates. It was quite a lot. And once, after 8 days, once I had my plates, so in that case, what I did is I measured, like I said, the distance of the fruiting bodies that had developed away from the starting line. And I call these the migrators. But there were also fruiting bodies that had developed right on the zone where I had loaded the samples. So I call these the non-migrators, and their distances is 0 centimeters. And I also collected the spores that these fruiting bodies made, both for the non-migrators as well as the migrators. So what would my hypotheses look like for if I were to find these so-called unselected incompatibilities in development? So first of all, my null hypothesis is that if you mix the ancestors with their evolved lines, the average is what you would expect. There is not much difference. It's not better or worse to be mixing for the developmental trait of slug migration or spore production for that matter. But for the case of unselected incompatibilities, these organisms that have developed from a mix of their ancestors and these evolved lines, which have sort of different genetic backgrounds, they should, if mixing gives rise to incompatibilities, they should have worse performance for the traits that I'm measuring, right? So mix should be lower than the expected average of ancestors and the evolved lines. And when I saw the results, that was quite interesting, but in a not interesting way. So on my y-axis here, I have average slug migration distances. My x-axis are the treatment. And remember, I said that my expected value for the mixed lines is the sort of linear average of the ancestors and the evolved. And I found no sort of difference between them. In fact, it looked very jarring. So then I measured whether this is a linear term or a polynomial sort of coefficient can fit this regression better. But I found that this is very linear. So it lies right at the middle of the ancestors and the evolved. Okay, so as you can see here, the quadratic term doesn't make sense. Then I wondered whether it's because there are just more migrators for the ancestors and less migrators for the evolved lines, such that I just have less of a sample size to be measuring from. But interestingly, that wasn't the case, because there is no sort of difference between migrators or non-migrators at the level of treatments here. They are all, the proportioning between whether you migrate or not is the same and seems to not matter. So then I looked at what happens when you measure the spores of these migrating fruiting bodies versus non-migrating fruiting bodies. And this was interesting, but in a surprising and interesting way, not uninteresting way. I saw here that the mixed lines actually produce significantly more spores, not just from the expected average, but also from either of their ancestors or evolved counterparts. And that made me go back to my plates. And this time I made a mix of the evolved lines that had diverged with each other for a particular clone, and then measured the same thing of their average slug migration distances. And guess what? I again found that there wasn't a difference between whether you are an unmixed fruiting body or whether you develop from a mixed sort of propagule there. So that tells me that there's not that much of a cost of mixing together, either for the evolved dictyostelium lines, whether they mix with their ancestors or other evolved lines. So it seems to be that there's not many, like, unselected incompatibilities are not really likely in this system. And with that, I would have some conclusions, which is that I don't really see any evidence for unselected incompatibilities in dictyostelium. And slug migration, the one that I showed, is sort of very on the point of being a linear trait. It's actually an additive trait, so there are not sort of probably interactions going on, which is the case for spore production, where if you remember that it was sort of better, but that doesn't necessarily mean that development or fitness is an improvement in this case, because dictyostelium also needs its stalks to raise higher from the soil surface and be dispersed into newer locations. So depending on the environmental conditions, it might actually be worse to make a lot of spores without having enough cell real estate to be making stalks. And as you can see, I thought this is cool, but obviously someone else 20 years ago had done something similar and had found some useful things for me. And this is that, so Kevin Foster, who had measured slug migration distances for clonals and different strains of wild dictyostelium. So these are collected from the wild, and they have had enough time to sort of develop their incompatibilities, but they are not of the same strain. These are different natural strains. And he found that when you mix these sort of genotypes together, you get shorter slug migration distances. But at the time, something that wasn't really known was that cells in a dictyostelium slug can preferentially adhere to each other based on these genes that give them the ability to discriminate between self or non-self. So it could be that the incompatibilities that we do see in dictyostelium are a result of evolved incompatibilities, and these non-selected and non-adaptive mechanisms seem to be less relevant for Dicty's own development towards aggregated lifestyle. And with that, I would like to give thanks to my lab mates and the Queller-Strassman lab, especially my advisors, Dave and Joan, who are here if you have major criticisms about this. Also, I would like to say thanks to Debbie, who taught me a lot of the Dicty culture that I learned and has retired now, so farewell. And I would also like to thank my funding sources. Thank you.
Okay, we have time for questions.
In many other organisms, the kind of incompatibilities don't show up in the initial hybrids, but rather only in back crosses or F2s. So do you have plans of continuing these further generations?
Oh, no, not at all. I don't have plans to evolve these, but hopefully that can be done by someone else. But I'm not, so when you say F2s, do you mean particular, let's say 10 generations, 30 generations, like 50 generations? Sort of
Generations.
These are asexual, just cell divisions.
Okay. Is there any mechanism for genetic recombination in these? Because that's usually when these incompatibilities show up.
Yeah, so I mean, I'm just using that as an analogy. And dictyostelium does have a sexual cycle in the wild, but it's not something very easily accessible in the lab.
No.
Yeah, but it would be, yeah, I'm you're right about the initial conditions having they don't show up much, but over time. And in fact, I thought that that's why I had done the later experiment of mixing these ones where they could have parallel sort of alleles that are not encountered. But even then, I didn't really see very much.
Why is slug migration distance a measure of fitness?
Because did I not mention that? Oh no. So it's like I said, it's beneficial for slugs. They are living in soil, eating bacteria, but when they need to be dispersed, they are dispersed by small invertebrates or birds and other, herbivores and stuff. So they need to come up to the soil, a surface, and that's why they are moving towards light. So it is, and it has been known, like there are evidences showing it's, if you travel farther, it's better. You have better chances of dispersing to a food-rich condition because they're making these slugs when they are in a food-poor condition. Yeah, so it's to escape misery. Okay, thank you, everybody. I'll switch your slides here. I'll put that on. Mm-hmm. All right, there we go. All right.
Where did it go?
Up next, we have Eduardo Aguirre-Mazzi from who hails from La Paz, Bolivia. He completed his undergraduate degree in molecular biology in 2015 and his master's in biology in 2020, both from Universidad Mayor de San Andrés. He's currently a third-year PhD student, co-advised by Christine Edwards and Sebastian Teo. He's currently sponsored by the Catherine M. Lineman Scholarship, and when he isn't doing science, I happen to know that Eduardo plays in an Andean folk band. So let's give Eduardo a warm welcome.
Right. Thanks, Anna, for that kind introduction, and thanks everybody for being here. So I'm going to present about well, you already know the title probably. Yeah. Phylogenomic insights into the evolution, biogeography, and taxonomy of Andean Weizmannia, corresponding to the family Quinoniaceae. So this work that I will present here, will be part of my thesis proposal that I will be defending in a month from now. And so the results I'll show now are by no means like conclusive, and they're more like exploratory or preliminary in a sense. So, there we go. So first of all, Weizmannia is a diverse genus of trees and shrubs. They're considered like they're the largest genus of the family, and about like 75 to 90 species have been recognized in the Neotropics and 2 species in the Mascarene Islands. And this genus has typically small fragrant flowers that attract bees and other small insects, and they form this paniculate inflorescence. Their fruits are, as you can see here, are like small, hairy, dust-like that are easily dispersed by wind. They're characterized by displaying a high level of phenotypic diversity, and particularly in the Andes, there's a lot of species. They range from like simple leaves to compound leaves, going through sort of these intermediate auriculate leaves with varying degrees of trichome density, that means pubescence, and also leaf or leaflet sizes. So all of this morphological complexity leads to a complex and unclear taxonomy as well, as we can infer. So some aspects about the conservation of these plants. In the Americas, the forestation of mountain ecosystems are the main threats to Weizmannia populations. And as we can see here, out of 49 species evaluated for the IUCN, 16 fall under a threat category, including 3 endangered species, including, say, Weizmannia costulata from Ecuador, for instance. Just sorry. So in the Neotropics, Weizmannia mainly occurs in mountain forest ecosystems, and mainly they occur in a diverse range of mountain forest types through the Andes, but also in the Central America and the Caribbean islands, and also in the Tepuis Formation of Venezuela here. But we can find also some species in the Atlantic forest in Brazil, and we also find species in the Valdivian temperate forest in the southern cone of South America. However, in this place they occur at lower elevations because of temperate weather. So there are like 2 main broad hypotheses to explain origins of neotropical flora. One of them poses that lineages might arrive from boreotropical locations and from North America, basically, and they might disperse southwards. This suggests a Laurasian origin of these lineages, whereas alternatively, they can have arrived from derived from Paleo-Antarctic lineages, suggesting Gondwanan origin of these lineages at a deeper scale. So in addition to historic processes, I'm also interested in more biological phenomena that might determine the diversification of these plants. And for instance, it has been observed that sympatric species tend to have distinct morphologies that correspond to local habitats. And as we can see here, well, in this, here we have elevational ranges for these species that coexist within the Madidi region in Bolivia. And as we can see, these species sort of partition elevational niches in these mountains. So even though when they coexist at the same elevations, they tend to have distinctive morphology, such as the ones shown here with Weimania with compound leaves and small leaflets, and Weimania crassifolia with broad leaves. Big. It's a simple leaved plant. So this has this broad phenotypic variability that it's apparently associated with environments and elevational niches in these cases have led us to think that this might be a case of an adaptive radiation. So that's one of the things we are interested in testing. So now to test all of this, to understand more the biogeography and evolution of Weimania, we need phylogeny first. And very little is known about the phylogenetic relationships on this group. Previous work were mostly conducted at family level, including very few species of Weimania and based on limited amount of characters. And here we have some examples of phylogenetic work made on morphological data, traditional Sanger and fossil records, phylogenomic study at family level using Angiosperm 353. And more recently, our collaborator, Francisco Fajardo, is working on plastome sequencing and nuclei markers. However, in addition to that, multiple factors complicated phylogenetic inference for this group. Among them, we can mention pervasive hybridization, potentially rapid radiation, and mostly lack of comprehensive taxonomic treatment across the neotropics. Well, this last point mostly complicates taxonomy and systematics rather than phylogenetic inference itself, but it's related. So let's talk a bit about the hybridization in this plant. So previous researchers, taxonomists, experts have shown that these plants tend to hybridize a lot, especially when they have overlapping distribution in the altitudinal gradient. They tend to form hybrids, and sometimes they identify these intermediate morphologies, as I mentioned before, like sort of auriculate leaves as hybrids between, for instance, in this case, of a compound leaf species and a simple leaf species. So this suggests that hybridization might be common in Weimania. In addition to that, there are some sort of taxonomically questionable species taxa that have broad distribution and phenotypic complexity across this range. So this also suggests that there might be cryptic species that molecular systematics might help to find in this case. So the overarching goal of my research here is to enhance the understanding of plant evolution and biogeography in the Andes and in the neotropics using Weimania as a study system. And here is one of my favorite Weimanias, Weimania tomentosa, which has a lot of trichomes, it's very furry. So here are my objectives. For chapter 1, I plan to reconstruct a genus level phylogeny while accounting for introgression and hybridization. Chapter 2 will be related to the understanding of biogeographic origin of Weimania. And chapter 3 to characterize the radiation and drivers of diversification of Weimania. So for this purpose, our team has gathered more than 70,000 specimens across the neotropics in Chile, Bolivia, Peru, Ecuador, Colombia. But we're also planning to sample in Costa Rica, gather some samples from the rest of the range of the species. So far we have collected around 51 species taxa. And out of this data set for building a phylogeny, we have selected around 3 individuals per species per region. And hybrids are identified either through taxonomic analysis or either through preliminary molecular analysis and are excluded from this phylogeny in this case. And well, the assembly is prepared in iPirate. So yeah, I guess for those that are unfamiliarized with tuberat sequence seq approach, it's just like a reduced representation a reduced representation of the genome that is based on the use of restriction enzymes, and what you basically get, it's thousands of fragments of DNA for each species sequenced. In this case, we produce fragments of 36 base pairs that are sort of concatenated. And so with this concatenated matrix, we just built a maximum likelihood phylogeny using basic RAXML approach, maximum likelihood. We included 153 specimens, and the final matrix includes more than 4,000 loci concatenating with a missingness of 76%. And as we can see in this phylogeny, the species are colored by country of origin, and we have collapsed all the species, and in parentheses, we have the number of individuals per species included in this case. So most of these species did form monophyletic groups. However, a significant amount of them did not cluster into monophyletic groups. So for instance, we have Weidmannia phageroides, which groups in different clades along the phylogeny. The same thing with Weidmannia pinnata, another compound-leaved Weidmannia. Weidmannia baldwisiana, which occurs in around 3 clades in this phylogeny, and Weidmannia sorbifolia. These are just some examples. And so now the question is how to explain the non-monophyly of these taxonomic species. And well, the first thing might be hybridization and introgression, and recent evolution that might lead to incomplete lineage sorting. So these two might bias phylogenetic estimation, especially hybridization and introgression might be producing phylogenetic errors because of we use a model that assumes bifurcating trees. In addition, we might be facing a scenario where we might have a convergent evolution of similar morphologies in different locations and clades, thus confounding taxonomies. And this could be suggesting potential adaptive replicated radiations in this case. Regarding hybridization, with this data set, our collaborator here, Juan Penagos, is doing extensive analysis of hybridization. For example, here we have 5 species that co-occur in the Madidi region, and here is a structure analysis for more than 300 specimens. And each bar here represents a specimen, and colors represent genetic groups. If an individual has no more than 85% of probability to be assigned to a genetic group, we consider that to be a hybrid. And here we can see a significant amount of hybrids here depicted with these black bars. Even with an extended number of species within this region, our collaborator has found that hybridizations occurs among lineages regardless of the distance of the phylogenetic distance here in relation to our phylogenetic tree. So this has led us to think that this also could be happening for the Northern Andes and the rest of the regions. So the next steps to make progress in this chapter will be to further explore patterns of hybridization in other regions, such as Colombia, Ecuador, Peru, but also explore the importance of introgression in the evolution of Weidmannia and build phylogenetic networks that will allow us to obtain a more, to capture the reticulate evolution of the group, basically. So regarding the second chapter, understanding the biogeographic origin of neotropical Weidmannia, well, just by sight, we can see that the phylogeny suggests a southern temperate origin of neotropical Weidmannia here. As we can see here, for instance, species from Chile, Weidmannia trichosperma, and the southernmost species of Bolivia, Weidmannia boliviensis group, are placed near the root of the tree. We can see gradient of like sort of like a northward dispersal from south to north. So basically the lineage has diversified as it dispersed towards north. This observation is also supported by a high phylogenetic signal for latitude. And in order to understand better the biogeography of this group, we are aiming to get a time-calibrated phylogeny as well. And for this purpose, we have used previously modeled age, which is placed in the root of the tree, and also a fossil pollen record, which was found in the northern Andes. So the clade that contains species from the northern Andes were assigned to this calibration point. And this result shows that Windmania, neotropical Windmania, as we know it, might have arose 12 million years ago around. So using this tree, we have done like some ancestral characteristic reconstruction for latitude. And this is more like exploratory analysis, but we have built a model to predict the node age as a function of latitude. And as we can see here, the slope parameter for latitude, it's different than 0. As we can see from this posterior probability distribution, that suggesting that latitude it's a good sort of predictor for the node age in this case. And this result supports northwards dispersal from southern temperate region, and these sort of also suggest a paleo-Antarctic origin of Windmania. So Windmania might have dispersed to southern latitudes in South America before Antarctica frozen around 20 million years ago. And from then, it might have dispersed through the Andes, probably coupled with the latest uplift events in the Andes. However, we still cannot just like that discard long distance dispersal. But yeah, this is what we see. So next steps to move this chapter forward will be to include Windmania tinctoria and Windmania mauritiana from Mascarene Islands, and also include Windmania paulinifolia from the Atlantic forest. So this species will help to clarify the relationships in the base of the phylogeny, and also help us to get a better estimate of divergence times. And we plan to expand the sampling with species from Central America, basically southern Mexico and the Caribbean islands. And also, well, incorporate Windmania potosina, which is a fossil that we just found here, and we plan to introduce. Well, we didn't found it. I found the study that cites the fossil, and also do more like biogeographic history reconstruction in a more rigorous way. Finally, I'll talk a bit about my last chapter, which will be sort of characterizing the radiation and drivers of diversification of neotropical Windmania. And sort of this is the chapter that needs more sort of conceptual development. So any feedback is appreciated. And so just to explore the evolution of some ecological traits, I did like a ancestral stage reconstruction for leaf type and also for elevation. Well, I don't want to get into the details of these figures, but as we can see from the leaf type reconstruction, we can see multiple transitions from a compound leaf, imperipinate leaves, to simple leaves across the phylogeny. So this transition might have occurred multiple times, the same for auriculate leaves. Putting all these results together, again, this is like, these results are more preliminary and generate more questions than answers. And here I show a phylomorphic space plot showing ancestral space reconstruction for elevation on the y-axis, latitude on the x-axis, and leaf type by colors. And big dots represent extant species, and small dots represent nodes in the phylogeny. And what I wanted to show here is that when we compare, like we have two groups here, the Central Andes and the Northern Andes, we can see how all of these species are sort of diversifying into the whole altitudinal gradient within these regions, and also evolving these morphologies repeated times, well, at least twice. So this, as I said, this could be a characteristic of replicated adaptive radiation. So now we're aiming to explore what are the main drivers of the diversification of these plants here. So for this chapter, we plan to measure like some ecological traits and identify ecomorphs, and some, just to put an example, we plan to measure leaf areas, trichome densities, and probably size and density of vessels out of herbarium specimens, and then correlate these ecomorphs with environments as a signature of adaptive radiation, and also test for convergent evolution of ecomorphs in areas of endemism. Just to conclude, I want to mention some key points and sort of future perspective. Of course, more work is needed to understand the evolutionary history of this group, and that might be beyond my dissertation, but yeah, and yeah, here we want to kind of we want to propose Weimania as a model system for understanding ecology and evolution in the tropics, in the neotropics, and particularly the pervasive hybridization and distribution across disjunct mountain regions could allow to explore relationships between sort of reticulate evolution and diversification and adaptation, that kind of thing. So this would allow us to explore drivers of diversification from a comprehensive perspective, integrating genomics, taxonomy, macroevolution, ecology, and with sort of potential implications for conservation. But maybe that's too broad to say. Yeah. Yeah, with that said, I just want to thank our vast network of collaborators practically across the globe, and the different institutions that have participated in this, that have allowed this research to happen, this data set. And also I want to thank you for listening to this talk, and I'm open for questions if there's time.
Yeah, I was really intrigued by your hybridization not being related to phylogenetic position. So do you think this genus is really a syngamion? And if it is a syngamion, how would that affect your interpretation of the work for chapter 3?
Right. I was hoping not to get that question. No, that's a joke. Actually, yeah, it's good. It's an excellent question, and I think it all depends on what is the concept of syngamion we're using, and we might have to revisit the species concept into that. That's a complicated issue to discuss, but we have a good evidence that these like sort of species might have a sort of phenotypic cohesion. Of course, the flowers are in general are ecologically equivalent, so I guess we observe that when they co-occur, they hybridize, but they also have sort of particular ecologies, or at least that's what the data seems to suggest. So we might need to revisit which species concept to
Right.
Yeah. I don't know if that answers the question. Yeah. Mm-hmm.
Hi, fascinating talks. Cool, thanks.
Thanks.
I will have a question about if you could comment on what the dominant dispersal mode is, and if there's any variation in it, perhaps how that dispersal mode might shape the relationship between node age and latitude.
Mm-hmm. That's a good question. Well, as I mentioned, the seeds are dust-like and hairy, so they're most likely dispersed by wind. But since they're also hairy, well, all of this is like there's no like studies that really address this like aspect of the dispersal ecology of Weimania, but based on the syndrome that the seeds have, they might be most likely dispersed by wind, but also they might attach to animals because they're hairy. They might attach to birds probably and disperse. But yeah, regarding how that might affect the dispersal, I think this is like more in the time scale we are talking about, it's like sort of 5 million years to 12 million years. So I guess they could just easily disperse by wind and progressively being, yeah. I don't know if that answers the question or want to follow up. Thanks. Okay.
I have a question about this clade. Oh, it turned on clade really quick. Is this are plants in this clade expected to have polyploidy? And if so, are the number of genomes that you would expect in a plant be affecting the types of analysis that you do, especially with your phylogeny and how you're interpreting hybridization?
Yeah, regarding the the first question, we have not really explored polyploidy, but through bioinformatic analysis in iPyrite, we can say that we don't expect them to be polyploid due to the like sort of it's a measure of parallel genes. So they're most likely diploids. And then regarding the number of you mentioned the number of specimens per taxa that we include for the, yeah, that's definitely a factor here. And that's why we focused our analysis, well, Juan is focusing his analysis mostly in the Madidi region where we have a sort of extensive sampling of species. However, when we go to Ecuador, we don't have as many individuals per species, so we might need to sequence some more to make further explorations of hybridization processes. Yeah. One more, yes.
When you were talking about chapter 3, you listed 3 traits that you're going to examine, 3 morphological traits.
Yes.
How did you choose them?
These are, well, basically these are ecologically important traits. For instance, leaf area might determine, it's determining the surface, the photosynthetic area. So in general, well, here I talk about leaf economics spectrum. And we can see, like, for instance, some conservative plants, we call them, they tend to have smaller leaves and high wood density, and they grow slow. Whereas sort of acquisitive plants might have, say, broader leaves or bigger leaves, and they have less wood density, and they tend to grow fast. And sort of it's sort of like a relation related with the pioneer or sort of primary forest.
Do you have any data from Miami that are important in that divergence?
Not really. That's what we are looking forward to explore. And, yeah.
Has anyone transplanted, sort of high altitude ones down to low altitude and vice versa? I'm wondering how much the environment contributes to the morphological diversity.
Yes, there are some studies that have done like these transplant experiments, mostly to see the thermal, what's this word? Well, how they perform in different temperatures. But yeah, it seems like they tend to have, even within the same individual, they tend to have significant morphological variation in leaf sizes. But I guess some other traits might just maintain, like their leaf architecture might not change within the same individual. But yeah, so that's why we try, well, at least I think they try to collect mature leaves always from leaves that are exposed to sunlight, so they tend to be more uniform.
Is there any other genus in this family? Like, could you use the distribution of other genus of the same family to infer the migratory history of this genus?
I'm not sure what you mean by other genus, but
This is a big family, right? And is it a single genus in this family or is it all genus?
sorry. No, there are like I think there are several genus in this family. However, they are mostly distributed in the Old World in southern latitudes, and in the Neotropics we just have 3 genus, but 2 of them only have very few species, and Weightmania is the most diverse genus in the Neotropics with more than 90 species. So I guess, yeah, a family level study would also help to infer how they dispersed into the Neotropics, but
yeah. So from the family level phylogenetic inference, the migratory paths from the Antarctica is preferred. Is a preferred hypothesis then from North America?
actually our collaborator, Francisco Fajardo, who's working in Germany with his PhD, he's kind of addressing that question. We'll have the luck to share some data on that, so it'll be interesting. Yes.
It's just such an amazing set of wonderful biogeographic samples. Is this herbarium and lab work, or did you get to do some of the field work? And if so, do you want to tell us some cool story about it?
Yeah. To be honest, I arrived in the lab when we already had most of these specimens, and it's a collaborative effort. And however, yeah, I kind of basically my work so far has been mostly sequencing some specimens, not all of these, and also doing the bioinformatic analysis and all that stuff. However, I'm planning to travel to Costa Rica next year to get some specimens there, and who knows if we can expand to other regions as well. Yeah, and then I'll have stories. Yeah, and I'll put pictures.
Well, let's give Eduardo one more thank you. Join us across the hall for refreshments and keep the conversation going.